Welcome to this lecture on fundamentals of genetics. Today, we are going to explore a fascinating topic, Ty elements in yeast. Imagine if paragraphs in a book could copy themselves and jump to random pages. That is exactly what these genetic elements do inside a living cell. We will uncover what they are, how they move, and why they are so crucial for genetic diversity and evolution. Let us dive in and discover the microscopic world of jumping genes. Now that we have introduced the idea of jumping genes, let us look at how they actually function. In the microscopic world of yeast, there are special pieces of DNA called transposable elements. Instead of just moving from one place to another, these specific elements make a temporary copy of themselves using a different molecule before pasting the new copy back into the DNA. This incredible copy and paste ability allows them to multiply rapidly and spread throughout the organism's genetic code, acting almost like an internal parasite. Building on the concept of jumping genes, let us specifically examine Ty elements. Ty stands for transposon yeast, and these are a specific type of transposable element found in common baker's yeast. They are formally classified as retrotransposons. Why? Because they move throughout the genome using a copy and paste mechanism that strictly requires an RNA intermediate. Because of this structural and functional similarity to certain viruses, scientists believe Ty elements are actually ancient relatives of retroviruses that became permanently trapped in the yeast genome long ago. Knowing that Ty elements are like ancient retroviruses, let us look at their physical structure. A typical Ty element consists of a central coding region that is flanked on both sides by long terminal repeats, commonly referred to as LTRs. The central region is highly functional. It contains two main genes that produce structural proteins and essential enzymes. One of the most critical enzymes produced is reverse transcriptase. This enzyme is absolutely needed to convert the temporary RNA intermediate back into DNA so it can be pasted into the genome. We have seen their structure, but you might wonder why yeast cells tolerate these genetic hitchhikers. T elements are actually incredibly important for the evolutionary adaptation and genetic diversity of yeast populations. When a T element jumps into a new location, it can alter the expression of nearby genes, or even completely disrupt a vital gene, creating a brand new mutation. Without these jumping genes, yeast would have a much harder time adapting to sudden environmental stresses. Furthermore, in biotechnology and brewing industries, understanding T elements is crucial because their movement can spontaneously change the traits of industrial yeast strains. Since T elements drive evolution, we need a way to measure their spread. Because they use a copy and paste mechanism, their numbers can theoretically grow exponentially over time, much like a population of reproducing organisms. We model this accumulation using a standard exponential growth formula. NT represents the total number of T elements present in the yeast genome after a certain number of generations. N0 stands for the initial number of T elements present at the starting time. The variable R represents the transposition rate, which is the probability that a single T element will copy and paste itself during one generation. Finally, T represents the number of yeast generations that have passed. If the transposition rate increases due to environmental stress, the total number of elements will grow much faster over time. Now that we have our exponential growth formula, let us apply it to a real scenario. Imagine a laboratory strain of yeast that initially contains exactly 30 copies of the Ti element. Under specific environmental stress conditions, the transposition rate increases to 0.05 per generation. We want to find the total number of elements after 10 generations. We substitute our known values into the equation. We add the transposition rate to the base value of 1, creating a growth factor of 1.05. Raising this to the power of 10 gives a multiplier of approximately 1.62889. Multiplying this by our initial 30 elements gives 48.86. Since we can't have a fraction of a DNA sequence, we round to the nearest whole integer. The final answer is 49 Ti elements, showing how rapidly the genome changes under stress. Having calculated how many elements accumulate, we must now consider the physical space they take up. When a Ti element inserts into a new DNA location, it creates a short, repeated sequence of the host DNA called a target site duplication. Therefore, every single insertion event adds both the length of the Ti element itself and the length of the newly created duplication. We represent this with a simple addition formula. L added is the total length added by a single event. LTI is the length of the Ti element itself, and LTSD is the length of the target site duplication. If either the element length or the duplication length increases, the total physical expansion of the genome for that single event will also increase. Let us use this addition formula to find the exact size increase for one insertion. Suppose we are looking at a specific Ty element that is exactly 5,900 base pairs long. When it inserts, it creates a target site duplication that is typically 5 base pairs long. We want to know the total length added to the genome by this single event. We simply substitute these numerical values into our equation. We add the 5,900 base pairs of the Ty element to the 5 base pairs of the duplication. This simple addition gives us exactly 5,905 base pairs. This number represents the standard unit of physical expansion for every single transposition event that occurs in this yeast strain. Now that we know the size of a single jump, we can calculate the total impact on the genome. Once we have that individual length, we can multiply it by the total number of insertion events over the organism's evolutionary history. This strategy gives us the total physical expansion of the genome. In this formula, delta G represents the total change in genome size. The variable I is the total number of new Ti element insertions that have occurred. Finally, L added is the length added per single insertion, which we just learned how to calculate. As the number of insertions grows over time, the total genome size, delta G, will increase proportionally, requiring the cell to spend more energy during DNA replication. Let us apply our final formula to see the massive scale of this expansion. Suppose a yeast genome experiences exactly 150 new Ti element insertions over its evolutionary history. We already calculated that each insertion adds exactly 5,905 base pairs. We want to find the total increase in the length of the yeast genome. We set up the calculation by multiplying the length added per single insertion by the total number of 150 insertions. Executing this multiplication yields a massive total of 885,750 base pairs. This absolute physical increase demonstrates how retrotransposons not only mutate individual genes, but also physically expand the overall size of an organism's genome over evolutionary time. Having calculated the mathematical impact, let us visualize the entire biological process. Here we see the tie-element retrotransposition cycle. At the top is the original tie element embedded in the host DNA, clearly showing the central TYA and TYB genes, flanked by the long terminal repeats. Following the first arrow down, this DNA is transcribed into the wavy RNA intermediate. This RNA is then packaged into the circular virus-like particle shown on the right, where the reverse transcriptase enzyme converts it back into DNA. Finally, the bottom arrow shows this new DNA inserting into a fresh location in the host genome, creating the target site duplication at the insertion point. This diagram perfectly summarizes the copy and paste life cycle we have been discussing. We have covered a lot of ground today, so let us review the key takeaways. First, tie elements are fascinating genetic entities in yeast that act as internal genomic parasites, utilizing a sophisticated copy and paste mechanism involving an RNA intermediate. This classifies them as retrotransposons. Second, by continuously duplicating and inserting themselves into new genomic locations, they serve as powerful drivers of genetic mutation and evolutionary change. Finally, as we proved with our mathematical models, this transposition process not only alters genes, but physically expands the overall size of the yeast genome over time. Understanding these elements provides deep insights into how genomes dynamically evolve. Great job following along.